applications of Gaussian processes, all in the context of digital twins. So if you've not heard of digital twins before, I'm sure people will tell you what it means. There's a bit of a disagreement about it, but the use of Gaussian processes in this field, so helping scientists understand real-world situations via models, is long established, going back to the 70s, at least with Tony Hagen, through the 80s, 90s. It's kind of one of the main areas that Gaussian processes first got to be used. So first of all, I'd like to have Professor Steve Meager from Imperial College London.
Excellent, thank you, Rich. And it's great to be here, and I imagine it's a fantastic summer school because for us, we make computer models of hearts, as the movies behind us indicate, and every few years we talk to statisticians and computational scientists, and they tell us about the latest new toy that they've made. And that might be discontinuous Galerkin finite elements, or it might be dynamic mesh adaptivity. And we always want to believe that their new numerical method is going to help us to run our programs faster, fit our models, and often it works well in a square and falls apart quickly when you put it into a 3D heart model. And I would say that Gaussian process emulators and Gaussian processes have been probably the one method that has worked as it said on the box and really delivered for us. So I'm a big advocate and fan. So the work that we do is looking at making computational models of the heart. And the reason we do this is that coronary disease remains a massive problem. And if we look at what people are eating and how people are living their lives, I'm going to put a good money that it's not going to go away as a problem anytime soon. And so the challenge that we have with cardiovascular disease with our patients that we have is that they're very heterogeneous, and that when we give them medications, they don't always respond the same way. And when we take away their medications, they don't respond the same way. So this is a kind of a very short summary of a large trial that was done where they took patients who had been originally diagnosed with heart failure. And these might be women after they've had pregnancy and had pregnancy-induced heart failure. They might be people who've had drug-induced heart failure following their chemotherapy. They've been diagnosed with having heart failure. They're now on heart failure medication. And 5, 10 years later, their hearts have recovered, and they want to go off their medication. They're taking a whole lot of medication every day. They don't want to. And when they took the patients off their medication, 40% of them relapsed back into heart failure, which is a bad outcome. And so the guidelines say that if you have ever had heart failure and you're on heart failure medication, you have to stay on your medication forever. But 60% of people didn't go back, relapse. And so we're not really identifying these individuals. Likewise, if we look at people who have been given their medication, 31% of them recover from heart failure when first diagnosed, but 70% of them don't. And so how do we better differentiate these different patient groups? And there's also different therapies we look at, like pacing therapies, where pacemakers are put on the heart. And these are expensive therapies. They cost about £26,000 upfront cost to put one of these devices in. And 40–50% of patients don't respond to that therapy. So there is kind of this need to better understand why patients have different outcomes and what's underlying their disease mechanisms. And there's kind of a lot of ideas here from genomics, about precision medicine, about that if we can identify what's causing this variability of response, we can kind of stop this unnecessary or potential harm being done to these patients. And then we really need to understand more about the molecular underpinnings, what's going on in these patients' hearts if we want to determine how to fix them better. And so what I wanted to do is say that what we'd like to do to solve this problem is use mathematical models. And I was looking up what was the first kind of example that I could find of someone using a mathematical model to understand something deeper about the system that we're studying. And this was, and pronunciation, as I'm from New Zealand, is not great, but here we go. Eratosthenes of Cyrene. And he was in charge of the Alexander Library, so arguably a proponent of big data. And he had the idea that the world might be a sphere, and he looked at a shadow of a pillar in Alexandria, well knowing when the sun was shining directly down a well at the same time in Sain or Aswan, and was able to then estimate what using his mathematical model, was then able to estimate what the circumference of the Earth was to reasonable accuracy. And so this is where we can take a mathematical model, we can take observations that we have which are potentially limiting, but allow us to understand something deeper about the system and infer things from it. And this is really what we're trying to do with our mathematical models of the heart. And so it provides a framework for formalizing hypotheses, integrating data, and testing those hypotheses. And so for us, when we're looking at the data we get, we never have a complete picture of patients. Our patients are people, and they don't like to be cut up. And so we have to take non-invasive measurements. We have to measure blood pressure, cuff pressure to tell something about the pressure in the middle of the body. We have to use MRI, which can give us nice images of the patient's heart, but only at an incomplete resolution, at 10 millimeter slices, and yet cells are 100 microns thick. But we can augment that information with physical laws, Fick's laws, Ohm's law, conservation of mass, conservation of energy, about known physiology that we can observe from animal studies that we've done previously or cadaveric studies. And we can combine these into our mathematical models as a framework for integrating data and then looking at how we can add value to clinical care. So what does our model look like? So we're very focused on the heart. We start off with a mathematical model of the whole heart, which is characterizing the activation of the heart. This is solving a series of partial differential equations, looking at how the activation wave spreads over the heart. The electrical signal at the whole heart level is then represented at the cellular level by mathematical models or ordinary differential equations, which represent the dynamic conductance changes of the proteins which span the cell membrane. And so we're now down at about 100 micron resolution here. The ions that move in and out of the cell then activate chemical reactions inside the cell. So we're now looking at calcium ions which have been released from cytoplasmic reticulum, which is a microstructure inside of the cell. Those calcium ions then diffuse. Calcium ions then diffuse inside the cell to bind to another collection of proteins. These are the sarcomeres. This is about 2 microns length. It's the standard unit of a sarcomere. These have 2 thick and thin filaments, which are made up of, again, hundreds of proteins. And they interact to generate tension and force inside the cell. That cell contracts, and it does so within a tissue matrix. So this is where we have a series of cells which have a preferred orientation. They are linked with collagen, which gives us a preferred stiffness direction. And the cells are connected together, so the electrical activation has a preferred conductivity direction as well. We can combine these cellular models of contraction with our anatomical model, as well as boundary conditions representing the pericardium, or the sac that holds the heart, and the connections of the heart to the other tissue around it, to allow us to simulate whole heart mechanics. Then we take those boundary conditions from the mechanics to allow us to simulate computational fluid dynamics, look at how the fluid flow is moving in and out of the heart. Now, this is a reasonably complicated model. The current model we have has about 120 to 200 parameters, depending on how we're looking at it. And each simulation that we do takes, on 512 cores, about 7 to 8 hours. So when you're thinking about, which I'm sure you've done during this week, you've done sampling. And every sample you do, one sample, you have to send that sample to Scotland, simulate it on the national supercomputer, and then download the results, which are sometimes gigabytes, post-process the results, and then you have your single sample. So MCMC is not going to help you here, was pretty much what took me a while to learn. It's very exciting, though. And so what we'd like to do with our model, we have this big framework. We've spent years, and I realize how old I am now, but it is kind of a couple of decades now, putting all of this together, and what can we use it for? So what the doctors can do is they measure cardiac function, and then they would like to make a diagnosis. But with the models, we have this kind of underlying information about the molecular dynamics, the tissue properties, what the loading conditions are on the heart, what the shape of the heart is, and how all of that goes into cardiac function. So we'd like to be able to understand more about how the information on the, this is confusing, on the right goes into that diagnosis. We'd like to understand what the molecular targets are, because this can then be used to inform therapy. So what we'd like is we'd like to make some measurements and then be able to infer or be able to know immediately what the mechanism is. Well, what we're offering is a little bit different to that. What we're offering is that in our models, we can propose a mechanism. We can say, we think this protein is no longer working. We can then simulate it so that it isn't working. We can then run our simulations, and we can then test if our predicted change in function matches what our observations were. So it is a forward problem where we can test an idea, but not necessarily learn immediately what that answer is. And again, this is where, if we can use inference, we can possibly turn this arrow around. So how do we go about using inference? And so this is from our pre-Gaussian process days. We took a cardiologist, this is my good friend Aldo Rinaldi, who's there taking measurements in a cath lab. This is where they're putting, we go into the cath lab, and my PhD students have the excitement of kind of real mathematical research. We have to go out and get the data. You put on a lead suit. They make a slight incision in the groin. They then put a wire through the arteries, up through the aorta, into the heart, and make pressure measurements. And then we get this time transient that we're seeing along the top. And so I think all the PhD students in my lab at least have to spend one day in a lab collecting this data. And we then guess the parameters. So what's our best guess for the parameters in our model? We then make our model. We spend 4,000 CPU hours, which isn't a disaster, but it adds up per sample, to run the simulations to get the same measurement. We then compare to see if these are matched. If they do match within some L2 norm, we accept them. And we say, that's great. We found the one parameter solution, and we can walk away. And if not, we revise our guess and go around. And so this has been that was our standard process before. And so we wanted to look at what are other ways to do this a little bit more efficiently. And so this is where we wanted to test to see if we could use a combination of Gaussian process emulators along with history matching to infer the mechanisms which were causing a problem. And so we started off with an animal model. And so we worked with a group in Oslo, and they had a rat model. And so we have a healthy rat heart. And then what we have here is they put a little piece of string around the aorta, or where the blood comes out of the heart, and they make that smaller to make a constriction that increases the loading on the afterload on the heart. So when the heart's it's pumping blood, it's doing so against a higher pressure. And so the resistance of the aorta is going to be higher in our mathematical model of the aorta. So we then made a mathematical model of this rat heart. So we had our cellular model. We had a model of the sarcomere. So this is how you have two proteins which kind of bind together, and when they bind together, they can generate force. We had a model of shape. So we put a bunch of rats into an MRI machine and measured the shape of the heart with an MRI machine. We then took those hearts and put them into another MRI machine to measure the diffusion of water inside those hearts to get the preferred tissue microstructure orientation. And then we could do, after all of this, a simulation of one heartbeat. And so this was our virtual rat heart that we could then look to see, can we use this virtual rat heart to identify what are the mechanisms in a healthy versus unhealthy rat. So we started off, and don't need to know too much about this. This is a mathematical model of the sarcomere, and we had 4 parameters that we chose to characterize how the chemical reaction caused tension or forces to be generated in the cell. We then had a mathematical model of the whole heart. So we have a model that has a passive stiffness, it has a resistance, it has a pressure, and then it has a kind of an atrial pressure. So there's 4 more parameters there. And the model was then going to have an output, and we were going to output pressure and volume measurements, because these are the kind of things we can easily measure physiologically in the clinic. And so from this, we had our 8 parameters going to our input, and on our output, we had 14 different values. And it was taking us about 4 hours per sample on the single-core CPU for our small rat heart. And obviously, this is not a surprise to this men here, that we trained a Gaussian process emulator to this, and it then took 1.2 seconds. And so we sell that as a 12,000-fold speed up. Happy to be criticized for that kind of wild statement. But it did mean that we could then do large numbers of samples. And so when you can do large number of samples, that really changes the way that you're looking at your research. So as a PhD student, each one of my simulations took 24 hours. And I'd do one simulation at 24 hours, stay up till 3 a.m. when I knew it was going to stop, and then restart it. And I played a lot of PlayStation 2 waiting for those simulations to finish. And it didn't occur to me the idea of doing a sensitivity analysis on less than a few parameters and doing a sensitivity analysis only with finite differencing. That was the only thing that was considered. Whereas when you can now do 1 million samples with your emulator, you can now do global variances. You can look at over a full space. You can look at the interactions between the parameters. And so we can now look at doing simulations of our model and looking at sensitivities for all 14 of the different outputs. How do all these different outputs vary in the context of all of the input parameters? And does it change between the control animal and the diseased animal? And the reason this was particularly interesting was this project was funded by Pfizer, which is a drug company. And one of their questions was, can we run our drug samples and our drug studies, if we're going to do them on animals, and obviously they want to be very effective, they want to be efficient for lots of reasons, can we run them on healthy animals, or do we need to make a disease model animal, which is more expensive and worse for the animal? And so if you do a sensitivity analysis and you say, well, if the output you're interested in has the same sensitivities to the different input parameters you're interested in the diseased animal and the control animal, then that's possibly going to be fine. Whereas if the parameters which are driving the outputs are different between the diseased animal and the control animal, then you're probably going to need to use the diseased animal when studying your drug. So here, we could see that the end-diastolic volume, if that was your target of interest, which is a number for the volume measurement in the heart, and the calcium sensitivity, which again, another one of the input parameters, is dominant in the control animal, and tissue stiffness isn't that important. Whereas in the diseased animal, the tissue stiffness becomes much more important, and the calcium sensitivity is less important. So if you were making a drug which was targeting the tissue stiffness to improve the end-diastolic volume, you would need to do those studies on a diseased animal as opposed to the control. And so this was a way that we could start to show value for sensitivity analysis in the experimental design. We could also go through and use Bayesian history matching to calibrate the model. And we could look to see what the parameter differences were. And what was important for us was the Z parameter, which was the aortic resistance, which was how much was the resistance to the blood flowing out of the animal. We saw that the model was able to infer a significant increase in that resistance. So in this sense, in a very simple animal model, we could take animal data, fit our model to it, and identify the mechanism that we knew was driving the disease in this animal correctly using the simulations, using a combination of Gaussian process emulators and Bayesian history matching. So this gave us some confidence that the modeling framework we had was complex enough to represent the system, and that the approaches we had were going to be precise enough to allow us to differentiate between different mechanisms if there was a strong cause for that mechanism in the data we were collecting. And so what I wanted to do was also to kind of point out some of the challenges. So many people here might be just starting their PhDs, and some of the questions that we certainly have as users of this technology. And so we have, the heart simulations we have are complicated. They have a great nonlinear component, and almost all of it's nonlinear. And so we don't always have guarantees that the simulations are going to solve. And when your simulations don't solve, you have these failures. You don't know if they failed because the heart didn't contract enough, and so it didn't complete a full cycle, and so it was an unphysiological result, or that because your contraction may have become too fast, and the time scales that your solver or your numerical solver was running on became unstable, and so your solver fell over. So you end up with a large number of failures of simulation runs, and you don't know what's causing them, and you don't know how to deal with those when training your emulator. So for us, only about 20 to 30% of our simulations completed in this study. And then we had to build, and that's from our Latin hypercube sampling. You only get 20 to 30% of simulations running, and then you train your emulator, and you hope. And obviously, the numerics people in here, and I've been told this many times, say, well, shouldn't you spend a huge amount of time investigating why those simulations aren't running? And the challenge is that those simulations haven't been running for a decade, and there are entire departments of mathematicians looking at how they could be running better, and they haven't come up with a solution, and I'm probably not going to be the person to do it. So it means that there is a pragmatic decision here. And so that's a challenge that we have, and I'm be great if people have or research any solutions to this, because I think it would be an interesting problem to solve. So we now move from rats to patients. And so for patients, we can get information, but what this means is that as so I did my PhD in computer science, and then went into a hospital, and no one collected the data that I wanted. So now the lab has to run their own clinical trials. So we're running the Rhythmic trial, where we had to go out and get the money, design the protocol, do the ethics, and now have a clinician recruit the patients to get the data that we'd like to make the models. So we now have lots of pressure measurements, lots of volume measurements, lots of electrical measurements. So we have a great deal of data that we get from these patients. These require putting a lot of wires into patients' hearts and making extensive measurements. But this is what we're doing to make our models at this stage. And we have our model of our human heart that we can simulate all these different measurements. So we can get these generated out. And what's useful with this is it makes bright, colourful pictures, and that tends to impress our clinical colleagues and keeps them engaged and entertained. Also, pro tip, have them move. That's also a big benefit. So we have these models, and we'd like to calibrate them to the clinical data that we have. And so for our models, we have a model of the atrial myocytes. So these are the electrophysiology, or electrical properties and the mechanical properties in the cells in the atria, which are the top two chambers of the heart. We then have the same kind of models, the electrical, the chemical, and the mechanical models in the ventricles. We have a cardiovascular system, so our heart is not pumping against a vacuum. There is a whole model of the circulatory system, of the resistance from your lungs, the resistance from the blood supply all over your body that we have in the model. We have tissue properties in the atria. What's the collagen stiffness? How much does the tissue conduct? Likewise for the ventricle. We have regional differences even within the ventricles, the lower chambers, so we have different parameters in the right and the left side of the heart. And we also have this pericardial boundary condition, or the sac, that's constraining the heart. Because again, the heart isn't contracting in a volume either. And so this gives us about 120 parameters. And 120 parameter space is quite large, actually. That when you're trying to sample it, you do anything, 2 to the power of 120, it's large. It all ends up bad. And so we were looking at how we're going to calibrate a model of this size. Even if you have to train an emulator, doing 120, maybe do 10 simulations per parameter, maybe 20, but even doing 1,200 or 2,500 simulations is going to be expensive. Each simulation for this, we worked out, was about £4. So if you're doing 1,200 simulations, then you're 4,000, 5,000 pounds in per patient. So making a computer model for a patient becomes a £5,000 diagnostic test, which is more expensive than an MRI, a PET MRI, genomics. It becomes an incredibly expensive test. It's completely unviable. So what do you do? So what we want to do is we want to break this problem down. So when we looked at the models we have, the models of the cell are ODEs, and ODEs are cheap to solve. And so this kind of represents a kind of a schematic or picture of what the model of the cell looks like. And bear in mind that we have this model with all these different colored channels here, IK1, IKR, INAK, all of those are proteins that span the cell membrane. Every one of those has a model, which is a number of ODEs, which some poor PhD student has spent their life creating, and then they've glued a bunch of them together to make the model of the whole cell. And in our simulations, we're going to have 4 to 8 million of these cells are going to be solved per heartbeat in our heart model. And so this has a grand total of, if we look at we're just going to assume that the kinetics for each of these proteins remains the same, and that the density is what's going to vary in the cell. And that means that we have 29 proteins and 29 densities of proteins that we might want to vary in our cell model. Unfortunately, 29-dimensional space is also large. So what we then did, we looked at what was the key output of our cell model. In this case, we know that there's a chemical signal, the calcium transient, which is going to be driving contraction. And we can then parameterize that in terms, or make a parametric representation of the kind of signal that gives in terms of its amplitude, its baseline value, how long time it takes to peak, how long it takes to recover. And we can do train an emulator to our, this is a Gaussian process emulator, train that to our cell model outputs, and then do a global sensitivity analysis on the cell model for all the different outputs. We can then rank the sensitivity analysis and find which of the parameters explain 90% of that sensitivity. And then that gives us a ranked list of the parameters that we're going to be considering are important in our model. And we can drop off, or we choose to drop off all of the parameters which aren't explaining 90% of the sensitivity. So this means we then go from 29 parameters down to 8 or 9 parameters for the cell model. We have the problem that when we do these simulations that we make our cell model and we do a simulation with it and it generates a result which is implausible. It's not physiologically viable. So you have a tension transient and it doesn't generate anywhere enough tension to cause the heart to pump blood. So that is just not a viable space. So we don't really want to be looking at the sensitivity of the parameters in unphysiological spaces. And so what we then do is we add in a layer of history matching. So we take our initial values We train our Gaussian process emulator. We generate our test samples. We then look at what are physiological or non-physiological or implausible or non-implausible parameters. We enrich that space and then take new test samples and train against those, retrain our GPE, and then repeat the process again and again. So we can zoom in onto a space of physiologically plausible parameters. And so what that looks like here is we have multiple waves, and we have all of the parameters that we're interested in. And as we go through each wave, we then move from the blue, which is the highly varied space, and many of those are not viable, down to increasing narrow-bounded parameters or spaces or regions of the parameter space, which generate physiologically plausible parameter sets. And this is what we want to use to reduce the parameter sets that we have, and so that when we sample from a cell model, it doesn't produce a cell model which is never going to be able to run a full heart model, so we're now going to reduce the number of failures that we have in our full heart simulations. And as I said, there are a number of systems, and so we then have a process. And so here we have our cellular dynamics for the atria and the ventricle, our tissue physiology, our passive mechanics, and our circulatory system. We then perform multiple global sensitivity analyses along each of these different cell features. We add in multiple layers of history matching, and this reduces our 120 parameters down from our big system down to about 40 parameters that we want to evaluate in our 4-chamber model. So all of these simulations along the top, all of GCSA, they're all done on low-cost sub-modules, and this provides a way of doing a multi-scale filtering of parameters so that you can get rid of the parameters, as many parameters as possible using cheap simulations, and only focus on the really important ones for the very expensive simulations. And then we can generate these, which we need to work out a better way to present this result because it remains an incredible amount of work and looks very unexciting. But this is our sensitivity analysis for the input parameters and the output parameters of the model. So we can tell a doctor, if you tell us any of the volume or pressure measurements that you've taken of the heart, we can tell you which of the parameters in our model are going to be most important for that output parameter. And so what's nice is that we also have these particular parameters all map onto genes. So if someone says we have a patient who has a particular genotype, they have a mutation in their troponin C gene, we can say that that patient is most likely, here are the outputs that you should be measuring from that patient, which are most likely to be affected by that gene mutation. So it provides a way of making a map from the cellular scale and the protein scale all the way through to the whole organ scale and making a way to read between the two of those. And we can also do some tests. We can look at some common diseases just to make sure that this kind of thing is working. So for us, we had atrial fibrillation as a very common disease in patients. It's where there's a chaotic activation of the top chambers of the heart, but it's associated with a reduced atrial contraction. And we can look to, and this is due to a change in the L-type calcium channel. And we can look at, as a surrogate measure of contraction, the end systolic volume, so the volume, how much does the heart contract by, and we can see that we correctly pulled out the L-type calcium channel was on the list of parameters that were important. And we can do the same for the increased stiffness that we see based on the maximum volumes. That's how much does the atria inflate. That was also determined by the stiffness values. And we can do the same for parameters in heart failure. And so, in summary, what we can do is we can take predictions from the model and say, these are the things we think should be important. These are the things we think are sensitive. And we can go back to literature and say, is that supported by any literature evidence to provide us confirmation that what we've done is correct. This is always a validation step rather than just reading it off the shelf. And I think this is really the challenges that we have is that the summary here is that it took about 1.3 million core hours to ignoring the single cell model cost, so just for the big simulations for one heart. And computing cost keeps going down, but 1.3 million is a lot. And there are a lot of patients with heart failure. So the idea is that if we wanted to roll this out as some type of assay where we make models of patients' hearts, and in the digital twin concept, update those models through time as we get more data, our current approaches are not going to work. And the worst thing is, this was quite a sophisticated approach. This is quite novel. We were the first group to kind of do global sensitivity analysis on this scale of model. So how you take these type of sensitivity analyses and do them not on one patient, but on 10,000 patients is an interesting problem. And I think many of the modeling problems we see are making a Gaussian process emulator for the oil rig, the factory, the example test plane that I have, And the question that I have is, what if I need to make an emulator that's going to work across 10,000 models, which is specific, or through one model through time? So how do we make those type of emulators? One of the other things we've been interested in is looking at, because we are looking at patients, and if we think about many engineering applications, there is the CAD model, and there is kind of one CAD model for your Boeing 747, which shows how old I am, Airbus, well, anyway. And humans, there's a lot of variation of shape. So how do you look at how sensitivity in your domain impacts the sensitivity of your outputs? And so the way that we did this, and the reason we're interested, we took a CT image of a patient, this is where they have a whole lot of x-rays taken, they rebuild that into a 3D image of the heart. We could then build meshes of 20 different patients' hearts who've had these CT images. And we then wanted to quantify how does that shape that's varying between different patients, how does that impact the results? How do we do a sensitive analysis on shape? And so when we look at the images, they have about 1.4 billion values in them. And so doing a sensitivity over 1.4 billion values, we couldn't do that. So we then did a segmentation, and we labeled about 40 million voxels or pixels in 3D in the heart. But doing a sensitive analysis of 40 billion values was too many as well. So we made a mesh. That brought us down to 3 million values. We could then look at make a statistical shape model, and that brings us down to about 10 to 15 models, 10 to 15 values, with a series of modes which describe the shape. So now what we can look at is saying, how important are the modes of shape for describing the variance in the output? And so we can look at what these different variations look like. So we can look at the first modes. We can look at how they shape varies with these different modes. We can then look and see how much of the variance is explained by different numbers of modes. So in our case, we could take the first 9 modes, and they explain 90% of the shape variance in these patients' hearts. And so the way our sensitivity analysis worked was we could take our mode 1 and mode 2. This is, obviously, I've projected our 18 dimensions down to 2 dimensions. And we could take a sample. We generate lots of samples using the Latin hypercube sampling sequence. And for each one of those samples, we could generate a mesh. And then we could perform a simulation of that mesh and how that functioned. And then we could output from that simulation with that particular sampling and the shape space, the pressure volume loops, the electrophysiology outputs. And we could make a series of phenotypes or kind of features which we've derived from those values. We could then project those features back into our shape mode space. And so now we have shape modes going onto a series of features. And this is then where we can train our emulator to go from our shape modes down to our features. And what this then allows us to do is to look at the sensitivity of the shape of the patient's heart in terms of those modes on those output functions. And that allows us to then say, if we have a patient who has a particular shape, which type of function is that shape most likely to impact? So it really provides a nice way of systematically linking shape through to function in our simulations. And so, again, I wanted to highlight some of the problems we have, and I'm very happy for people who are doing PhDs to be like, that's an interesting problem. We have to remesh every time. So because some of the modes are very different, we end up with very different shapes, we remesh. So part of sensitivity is the shape, part of sensitivity is the remeshing process. We might need different meshes to solve, different resolutions to solve the PDEs. So for big hearts or small hearts, we might need different results, and that leads to simulation failures. And that we've currently only looked at healthy shapes, and we really need to expand this to a lot more modes, potentially capture the full variety that we see in pathological cases. And so that makes it increases the computational cost of these problems. One of the other areas that we're interested in is doing in silico trials. And so one of the cases we have here is looking at patients who have a healthy heart activation. So they have a rapid activation of the top 2 chambers indicated by the light green arrows, and then a rapid activation of the 2 main chambers of their heart indicated by the big green arrows. And some of these patients have something called left bundle branch block, where one of those green arrows fails, and so the activation of the heart becomes asynchronous. Right side of the heart contracts, left side of the heart doesn't really, and then slowly contracts later on. So it leads to an asynchronous, inefficient contraction. We then have the ability to put pacing leads into these patients' hearts. These are where electrodes are placed in different regions of the heart, and these basically return the heart to a synchronous electrical activation. And you can place those electrodes in the left ventricle as with conventional CRT. You can use new leadless technology to put them inside the heart. And then there are increasing numbers of different pacing systems available. So we now have 4 different potential modes of delivering this therapy. We used to have only one. So there's a question of how do we choose what therapy might be best for each individual patient. And so for this, we made models of a whole lot of different patients' hearts, and then we could go through and simulate their activation patterns and look to see which of the different therapies are best in each different patient. And again, this is where we have this large variation of shape. We have different therapies, and this is something where we could potentially use emulators more effectively. And our problem is with these type of things is that the simulations are very expensive to do. Again, currently we're only sampling real hearts, and so one of our questions is there a way that we can synthesize hearts that might augment this to find more extreme values? Can we find better edge cases? How would we do that? How would we confirm that? And can we identify rare events or rare failure modes that weren't necessarily represented in the original data set? So these are some of the kind of, maybe not Gaussian process questions, but questions that we have and are currently exploring. The last thing I want to talk about, or I think this is the last thing, was about how do we scale these ideas up from making models of a single patient to being able to make models of large numbers of patients. So for people who don't know, we have the UK has one of a very arguably world-leading, definitely world competitive biobank resources. So the UK Biobank was 1 million people who went out and had a whole load of tests done on them, genetics, what have you. And for us, what's interesting is about 50,000, should be up to 100,000 eventually, have had MRIs. So they've had a magnetic resonant imaging, which is a relatively expensive procedure to have done, but that's been done of their hearts. They also have had 12-lead ECGs, so there's 54,000 of those people have had an ECG measurements. So there's lots of data there. It costs, in case people care, about £9,000 for 3 years of data access to download it, which for some people that will be a big number, and for people who pay £1,000 for an MRI, it's a very reasonable number. So it depends where you come from. And what we wanted to do was look to say, these measurements have been all recorded independently, and obviously the big data people are trawling through this, as big data people will, and using neural networks to make inferences and what have you. But we wanted to think, is there a way that we can combine the data that was being recorded? So the ECG, which is measuring the electrical properties of the heart, the MRI, which is measuring the shape of the heart, can we combine those using models to learn something more about these patients? And so in particular, we have this ECG, and this is something you will have seen on any kind of medical show. And so you can impress people. The P wave is labeled there. That's when the atria activate. The QRS complex, which is labeled in blue, that is when the ventricles activate. And then the T wave at the end is when the ventricles relax. So you can impress friends and family with that doctor knowledge. But this QRS duration is going to be determined by those proteins which are spanning the cell membranes. It's going to be determined by the tissue structure. It's going to be determined by the shape of the heart. It's going to be determined in part by where you place the electrodes on each person's body. So larger people have electrodes often placed in different positions. Women who have large breasts often have the electrodes placed in different positions. And so how those electrode positions impact the measurements you're getting becomes quite important to consider. It might be important for the factors. And then we also have the tissue structure of the heart, as in what are the location of the structures that are activating the heart? How are they positioned, and are they important? And so what we want to do is make a mathematical model which could capture this. So we had our MRI from the data set. We could make a model of the shape. We could do electrical simulations on this. We could put that heart inside a virtual torso and then simulate the locations of all of the ECG lead locations, and then generate and simulate a QRS complex. And the problem we had, and then from doing this, we could then, if we want to use inference, by comparing the QRS complex with the actual measurement that was made in the patient, we should be able to infer tissue-level properties using this system. Now, the difficulty we had was when we made these models, we made a mathematical model of the heart, which had where all the activation sites were. That gave us about 20 parameters. We made a model of the ECG, which had all the lead positions. That gave us another 30 parameters. So we had about 50 parameters that we wanted to calibrate from almost no observation. And so this was not a great place to start. So we wanted to say, which are the parameters that we're most able to infer from the data that we have? And what we did was we took 4,000 patients. We looked at what their BMI and their age and their gender was. We could then do a latent hypercube sampling across that domain to find 10 representative patients across that whole space. We could then make mathematical models of those 10 patients, and we could then train emulators for each of those 10 patients on the 20 parameters which determined how we got a curious duration from our individual cell. And we could see that of those 20 parameters that we needed, most of the tissue properties, or most of this value we're interested in, the curious duration, was determined by the conduction velocity. We could then repeat the same process with our measurement, a model of our measurement system. This was another 30 parameters. And again, nearly 70% of the information, all of the variance was determined, or sensitivity was determined by the conduction velocity. So what we had was a model which was likely to be greatly determined by was likely to be able to allow us to infer what the conduction velocity was in each of these patients. So we could then use UK Biobank ECGs and curious durations. We could look at simulating our activation patterns with a conduction velocity as our sole parameter. We could then create the anatomies for these. We could automate this process. We could create an anatomy from an image in 8 minutes. We could then calibrate these to get the estimation of the conduction velocity in a total of 15 minutes. And that meant that we could do 100 cases a day on a 64-core machine, which seemed like a good number until you get to 50,000 cases, and now it's going to take you a year and a half on a machine. Obviously, it scales trivially, but it's still, even at 100 cases a day, 50,000 takes a year and a half. And so when we think about these kind of large population data sets that are out there, I have friends who are working on 1.6 million ECGs from Brazil, and this doesn't scale still, even at this speed. And so there is this kind of moving these complicated models, or even relatively simple models, into that massive data space is still a challenge for simulations, I think. But I think there's some interesting things that we can get out of this. So what's nice about UK Biobank is it takes these ideas which have been often solved on single models for one patient and really been studied to death. And you see a lot of papers in our community of people who did 1 patient, 3 patients, 5 patients, and they make bold claims. And we were then, well, what if we make 3,000, 4,000 patients? And when we do that, we can start to look at differences in gender. We can say women tend to have shorter curious durations. Is that because their hearts are, on average, smaller, because women are smaller in heart scale with size, or is it because their tissue properties are different? And if your tissue properties are different, then maybe you're going to respond very differently to a drug, whereas if your heart is just a different size, then you're probably going to respond the same to the drug, but it's going to be somehow affected by the heart size. And what we were able to show was that when we looked at inferring the conduction velocity, we saw no difference in the conduction velocity between men and women, and that all of the difference in the ECG could be explained by heart size alone. We could also look at people of different body weights. We could also look at people with different ages. And we could also then go into the data set and look at people who had heart failure and validate the results to show that we saw pathological conduction velocity values in patients with known diseases. And so this was really where we were starting to think about how you can take these models, scale them up to a point where they can be run at 50 minutes per case, which is not a disaster, and then they could be used as a diagnostic tool. And they could be updated regularly with information that might come from a wearable, like a ridiculous smartwatch or what have you. So I think this is really where we see that we are with digital twins, and that there are people who are doing digital twins where they have fantastic kind of closed systems, where they have embedded sensors in their planes or in their buildings. They then go to a cloud system, they get processed, and they go back. In healthcare, that's really challenging, because taking data from a patient and putting it into the cloud is an ethical and legal challenge. It is not a small deal, and when you want to do that, what you find yourself doing is just spending an awful lot of money. Once you have the information on the cloud, you can process it, and that's fine. And if you never tell anyone about the results, that's actually the easiest option, because you can put your data into a secure research environment. That's kind of a government-registered, legitimate thing for having a free space to do research. If you want to take information out of a secure research environment, that's a big problem, because that doesn't sound very secure to me. So you then need to create a bespoke system. So now if you want to create a digital twin, you have to take the information from your patient, put it into a bespoke, secure cloud system, process the data, and then give it back to the patient. Or give it back to the doctor and then give it back to the patient. And so these will then become a whole load of workflows. So I think that what I would say to people is that there are huge opportunities in making digital twins of patients. I think there's huge value to be given there. I think more people are wearing wearables, more people are monitoring their own health. There's more data available. I think it's a great time for doing this type of technology. But I think there are lots of legitimate security, legitimate technical, and legitimate computational, particularly computational statistics questions, and how we're going to do that at scale, and how we're going to do that in a timely manner. And so, yeah, and this was also, if we say it's going to take a lot of time to do 3 years to do the 100,000 cases if they ever recruit all of them. If we have 1.3 million CPU hours for 45 parameters, how do we scale that type of idea to large numbers of patients? And what happens if we want to add shape uncertainty into this and other uncertainties? Everyone says, throw all of your uncertainties in, and Bayes statistics will deal with it. And our experience is that that's not always the case. So I think there are lots of challenges there. So this is kind of the idea that we've had, is that what we'd like to do is provide a clinician and arguably an individual with a mathematical model of their heart. They can then play their heart almost like a computer game, where they can say, what if I do exercise for a bit? What if I lose some weight? What if I stop drinking in the morning? Whatever those choices are. How is that going to affect my heart? Is it going to improve my cardiovascular function? And this is to say that there's a bunch of people. So to put this together, it comes out as 45 minutes of colorful moving pictures. And yet there's an awful lot of blood, sweat, and tears that goes into making all this work, and particularly the PhD students and the postdocs and our clinical fellows who are recruiting the data, and our collaborators. Thank you.
Thanks, Steve. Questions?
Pardon? Wholesale models. So there is. Yep. So I guess there's two levels of wholesale models that I have come into contact with. There is the ODE level, and those ODEs for us are basically scale trivially, in that you can put them on as many CPUs as you want. They often are run independently, and they're coupled with a PDE at the tissue scale. So the cell models we use will have 17 to 50 ODEs. They're all nonlinear ODEs. They tend to be solved with kind of CVOD inside the system. And then we'll have 4 to 8 million of those per heart that we solve just in the background to drive the tissue models. So the ODE type models, we are 8 to 10 orders of magnitude more complicated than most of the single cell models in our field. The other one was we're not more complicated, then you go to a spatially explicit cell model, and that's where someone's solving a PDE with like 1 micron mesh resolution, including the intracellular tissue, structures and the diffusion of individual or continuum representation of the calcium ions. And those can be very expensive. So for the ODE models, these are a ton of ODE models all glued together. Versus the PDE models, the complexity can be equally as complex.
Henry?
Yeah, I think a lot of us are Gaussian process enthusiasts here. I was wondering if you could give a little more information about the actual Gaussian process partners. Is that, are they kind of, have you noticed sort of, are they kind of continuous inputs you're using, and then making the Gaussian process of those?
Yep, so I mean, we are very much, we are the Gaussian process end users. So we have Scalar. Our goal, or our challenge had been initially, was to think about how do we represent our entire problem as a series of scalar inputs and a series of scalar outputs. And it's obvious to me now that obviously we can take our mesh and make a statistical shape model and convert that into scalar out and make that a scalar input. But that took us about 3 years to realize before we were confused about how we could represent different anatomies. And we thought, well, we'll have to train a new emulator for every single anatomy. And how does that work? So once we could represent our anatomies as a series of scalar variables and the modes of the principal components, then the entire all hearts that we have can be described as a vector of scalars. And so it becomes very easy to put that into effectively we're using a kind of a single variable Gaussian process and train that and generate our emulator. So I am not going to stand here and say that we are a sophisticated Gaussian process. We're not pushing the bounds on the envelope of that, but we have a whole lot of problems which we'd like to solve with Gaussian processes, and I'm delighted to provide kind of data sets to people. If someone wants 400 heart simulations, that's great. We can package that up. Or people say we have a different way of doing emulation which is going to be really robust for if your Latin hypercube sampling space is incomplete. Very happy to try that, but we're not kind of pushing the envelope on novel GP technologies. Yep. So I mean, the two applications that we've looked at have been, and I didn't present all of a lot of people. So basically, valve design. So we've had the cardiologist came and they said, well, we've been putting these artificial valves into patients, and the artificial valves last 8, 10 years, what have you, and the patients are lasting longer than their valves, and we now have to put a new valve inside the old valve. In a valve-in-valve procedure. And I'm like, this sounds bad. But anyway, that's what they're doing. And they were worried that when they deployed the second valve, that meant that the valve leaflets were going to then be forced open all the time from the original failed valve, and that was then going to block blood flow coming out of the heart in particular times. So they were then wanting to ask us, what are the anatomical features? What is the risk? What are the cutoffs for when we're going to have too high a pressure gradients? So these are the kind of questions we get asked about the electrics. For the mechanics, we get asked those questions about patients who have inherited cardiomyopathies. So they might have a mutation in their heart, and they want to know what that mutation is causing. And we also have that question on if we're going to put a pacemaker into the heart, how is the heart going to contract? And even some incredibly simple questions. If I put a pacemaker on the heart that's this particular size, what's it going to bump into when the heart contracts? Is that going to hit the wall? Is that going to cause any more damage? Or is it going to come loose, and you don't want things coming loose? So those are the type of questions that we get asked.
As students of cardiac population modeling, we have been looking into benchmarks of electrophysiology in which you were also part of it, and I think the whole of the community and benefit from it. So I wanted to seek your comments. Is it possible now, as we have already embarked upon the journey of simulating or creating emulators or calibrating our models? So can we try to benchmark the process of creating emulators, or can we think about ways to create benchmarks for means to calibrate cardiac models, and will it help?
I think the reason that we created, so for people who in this community don't know, is we're solving a large complicated PDE, and those PDEs have all been, solvers have all been implemented by very dedicated PhD students. And for those of us who are supervisors, we know that sometimes the PhD code is not always 100%. And so we wanted to create a standard community solution to a PDE problem and then we had, I think it was 14 different groups, all solved the same problem and compared the results and could then verify that everyone had implemented the PDEs in the correct way. And this was the way that we could then show verification that these solvers were all correct. And that's something that doesn't really exist that I'm aware of in the Gaussian process emulation kind of community of people's verifying that their implementation of their GPE is correct. I'm sure there are some analytic problems you can test against, but as things get more and more complicated, then there becomes a need for some kind of standard question that you can potentially answer to kind of give you confidence that the code's been implemented correctly. At the Turing Institute, we're currently working on a platform which would ideally allow you to use multiple emulators for a same data set. And then you could then compare multiple implementations from either the same GPE implementation or multiple different ones, see if you get the same results, and that might give you some confidence. But I think that if people can kind of come together and say, here is a standard test problem, it can really accelerate development of people who are trying to implement new code. So I'm all for those.
Hello, thank you very much. I've got a few questions, and also I'm on the periphery methodology applications. I've mainly methodology applications. How do you optimize?
In what sense?
Well, Gaussian processes depend on a bunch of parameters.
On the hyperparameters.
Yes.
So we will use a five cross-fold validation and a grid search.
So it's just an optimization scheme. It's not a multi-color scheme.
No.
And how do you check that the structure of the Gaussian process is appropriate for the problem?
So for that, we tend to use the, obviously in the validation, we look at the kind of correlation between the emulation and the holdout data and use that as a scalar variable for saying how confident we are in the GP. And then we also look to see how many of the points fall within the 95% confidence interval of the GP. And if that's 95%, then we tend to be happy. But I'm completely open to improve ways. And as I said, one of the things that I would like to see was lowering the barrier for people, naive users, let's be fair, to make better use of these technologies. So if there are better checks to do, then very happy to consider those.
One of the things that I've recently discovered, as much as I'm not confident, is that if you have, for example, the long correlation, the things is horrible, no matter how much you try to it will just not.
Yep, and I guess that's where we just keep searching. We have a very generous compute budget, and that has been our solution to many of our problems.
That's one of the solutions.
Oh, so I think.
I remember being quite surprised learning quite how much the NHS infrastructure runs on AWS.
Oh yeah. Well, you get to spend a good amount of money on AWS, well, or Azure. So both of those are viable options. I mean, within the NHS, so NVIDIA are putting DGXs boxes into the NHS, and so you can just whack a bunch of A6000s in there. And the last hospital I was at Tommy's, we had a couple of those systems set up for processing patient data. So I think the processing patient data in HPCs in hospitals is entirely plausible. I guess I also look at the desktop computers, and certainly AMD are pushing to 196 cores, I think. And you start to be like, well, maybe I get two of those or whatever, and I have a desktop with a couple of hundred cores. It might be able to solve most of the problems that I used to have needed HPC time for. So I can see that need for an HPC for a lot of the problems that we have diminishing as these kind of desktop computers are really scaling up the number of cores they have. And then for cloud, I think AWS and Azure and Oracle and whoever else is out there are likely always going to be part of it. Because as soon as you have people wearing wearables and they have to have that information uploaded somewhere, the NHS isn't going to run its own cloud server, and nor should they. So then you're going to be going for a low-cost, ideally secure, large commercial system, and that's what you can get with those big providers. So I would only say that for me, I'm afraid of the cloud. And I'm afraid of the cloud as an academic PI because we've had cloud time and we have lost thousands of pounds by leaving a GPU cluster open in Ohio and not being able to find out that it was still on and not being able to turn it off. And that happens once or twice, and you're like, this is a scary process to get that built. And so I think if there are safer systems and more budget-constrained systems, then that becomes less of a concern. But that's certainly, as a very naive user, that's something that was certainly a little bit alarming to see how quickly we could go through quite a lot of money.
About your multi-scale approach, you obviously rely on the understanding that people have of how the hardware and tissue and so on. That's not correct. People are still arguing about it in many ways. There's still some people who argue exactly what the mechanism is and exactly how. And so my question is, do you think that the effects of those kinds of uncertainty are bigger, on the kind of predictions, are bigger than, for example, the shape of the predictions? Are you worried at all about that?
I guess I think that often there are big arguments about very specific things. And so the macro narrative that we've presented here, I think it would be unlikely that there would be any physiologist who would say, I don't believe that calcium is driving contraction. That would be a bold statement and a tough platform to stand on. The specifics and the subtleties of does the crossbridge have a super relaxed off state? Does it not? Is this protein dominant or is that protein dominant? Those are subtleties which can be important, but I guess most of the models the cell models will replicate the experimental measurements that people have seen. And so they may have the mechanism not necessarily 100% correct, but the result in aggregate looks very similar. So some of the subtleties of what proteins are important might be different with different mechanisms, but I think on aggregate over the whole narrative, I don't think we're on any controversial physiology at this stage.
Does it work in a way? Does your work help inform where more research is needed?
Yes, I mean, I think we have one example where we're working again with a group in Oslo, and they had done a bunch of measurements, and they said, well, we saw this big change, and we said this particular protein explains everything. And we're like, can you model it just to kind of make figure 6 at the end and show that it all works? And we did that, and we said, look, you're missing. There's not the amount of ions you say that there should be, they cannot come through the system you've said. We've done the sensitivity analysis. We think that these are the proteins which are the top ranked. Can you look at the top ranked one and measure it to see if it's improved? And they went away, and it had. And that was actually kind of an important, not the dominant, but was a meaningful contribution to the story they were trying to tell. And so, in that sense, a lot of biology is, we saw a change, that must explain this. That might be it's a gross exaggeration, but a little bit. And the modeling forces people to write down all of their assumptions. It makes them write down all the things that they haven't measured and haven't considered. And it forces them to say, do the statements that I'm saying add up, even plausibly? And often they do, but in some cases they don't, and that's where I think the models provide a great way to give more insight.
One last final question then. If in terms of GPs, what's the biggest thing that's missing in terms of methodology, software, or theory, from your point of view, that make this work easier?
So I think I'll say that there are two things. So methodology or software that's missing is that we've tended to use a single GP library, and that has meant that we have been limited in the number of options. And I think the idea of having an ability to have a library of GPs and also neural network emulators or other emulators would allow us to kind of go through and make a comparison and make a choice as to which emulator was going to be best and which kernel was going to be best, what have you, for the data that we have. And that will be something that will be very useful. And for methodologically, I think that looking at how do you train GPUs that can then either be trained, if you have a large cohort of patients or models, how you train GPUs that can run, either be trained for low costs without doing too many simulations over that cohort, or how you can make GPUs which run and are relevant across an entire cohort of patients are the kind of questions that I'm particularly interested in. Because once you have that, then maybe you can make a patient-specific model without running any simulations, and that would drastically reduce the cost and make this kind of a viable technology.